All right, so here we are in chapter 3, which is the cell. This is talking about prokaryotic cells, eukaryotic cells, and some cell theory, the history behind the cell. Now, most of this chapter should be familiar to you. You should be already familiar and know this information. So I'm not going to lecture on the entire chapter. Instead, what I've done is just kind of a review. I've posted some lectures that I have created based on other texts that I've done in previous years. And I posted those up there. It doesn't follow along with the book in the same way, but just for your own use as a little reminder of all of these things. So I'm just going to kind of scroll through here and just say, so this is the beginning of the chapter here. The introduction, it's just kind of talking about what the chapter is going to have. So you should know spontaneous generation. You should be familiar with this concept of what spontaneous generation is, what the theory of it is, the different people that tried to prove it or disprove it. So that information there, spontaneous generation, and then this should be familiar to you from other biology courses. As we scroll through here, so talking about disproving it, as we mentioned. Right, and then foundations of modern cell theory. So the origins of cell theory, this should be familiar to you. We've already even talked a little bit about this cork photo that's here, but so that should be familiar to you and know that information. And here we are, the endosymbiotic theory. You should already know that, and you should familiarize yourself with that again, the information having to do with the endosymbiotic theory. And this photo should be familiar. Let's see, the germ theory of disease, that should be familiar to you. Refresh yourself on that information. And then what I would like to talk about is this chapter, the 3.3. So a lot of this is also, or should also be familiar to you from other texts, from other classes, but I do want to talk about this a little bit more because this class is microbiology, and we spend a lot of time looking at prokaryotic cells, a lot of time looking at bacteria very specifically, but then some other things. So I do want to talk about this chapter, so let's get into that. So the cell theory, which is talked about previously, states that the fundamental unit of life is the cell, but then they vary significantly in size, shape, and structure and function. So I'm going to go through some of this pretty quickly. This should be familiar to you already. So at the simplest level of construction, all cells possess these fundamental components here, including a cytoplasm. So you should know that these are the things that all cells share. So whether we're talking about a eukaryotic cell or a prokaryotic cell, these are shared by all of them. So cytoplasm, which is the gel-like stuff inside of the cell, contains or is composed of water and some other dissolved chemicals. The plasma membrane. So when we're talking about our cells, we're talking about cells that have a plasma membrane. Whether or not they have a cell wall, that's different, but they all have plasma membranes. Chromosomes, one or more chromosomes, so something that contains the genetic imprint or genetic blueprints of the cell, DNA or RNA, and ribosomes. So those are the 4 things that all cells have in common. So beyond these, then they vary greatly. So we have our prokaryotic cells and our eukaryotic cells, which are our largest categories of cells. Our prokaryotic cells, remember, are those that lack a nucleus, and our eukaryotic cells are those that do have a nucleus. So our prokaryotic cells generally have a single circular chromosome located in a nucleoid region. So rather than our eukaryotic cells here that have an actual nucleus surrounded by the nuclear membrane that contains these rod-shaped chromosomes. If you remember, rod-shaped meaning kind of like this, in this shape. That is in a eukaryotic cell. So we're going to mainly be talking about prokaryotic cells. So they do not have a nucleus. They do generally have a single circular chromosome, and then that's located in this nucleoid region rather than that kind of rod-shaped chromosome. So I'm going to scroll down just a little bit here so that we can kind of focus on our cell. So all plant cells and animal cells are eukaryotic. So we're not going to be talking in this section about plant cells or animal cells. They're eukaryotic. Some microorganisms are composed of prokaryotic cells, whereas others are composed of eukaryotic cells. So like a human, for example, is composed of eukaryotic cells. When we're talking about prokaryotic cells, we're going to be talking about bacteria and archaea. Prokaryotic microorganisms are classified within the domains archaea and bacteria, and eukaryotic are in eukarya. So the structures inside of a cell are analogous to the organs inside of a human body. So if we think about how the insides of the human body work, we all know we have a heart and lungs and our digestive system parts, and all of these parts work together to make this whole. Same thing we see inside of a cell. So some of the structures in a prokaryotic cell are similar to those found in some eukaryotic cells. Others are very specific to prokaryotes. Although there are some exceptions, eukaryotic cells tend to be larger than prokaryotic cells. The comparatively larger size of eukaryotic cells dictates the need to compartmentalize various chemical processes. So that is kind of lending itself to the idea that there's an actual true nucleus inside of a eukaryotic cell. So it's compartmentalizing those things. It's saying, okay, we're going to create a membrane and we're going to put the nucleus over here. We're going to keep the DNA in this package over here. And then we're going to have things like the Golgi apparatus and the endoplasmic reticulum, which are all membrane-bound organelles that are within the cell that are kind of compartmentalized. However, the prokaryotic cell generally lack these membrane-bound organelles. So they often contain inclusions that compartmentalize their cytoplasm. So they do have some kind of ways to compartmentalize things. And figure 3.12, which we're going to look at here in a moment, is what a typical prokaryotic cell looks like. So you should know these things here. So we will go into this in more detail, but make sure that you know this illustration here. This is the general form of a prokaryotic cell. Here is something that we've already talked about briefly, the nucleoid region, which we have our circular DNA. They actually sometimes also have plasmids, which contain DNA as well, or genetic information as well, and we'll talk about that in a moment. We do have our plasma membrane. Remember, that's something that's common among all cells, eukaryotic and prokaryotic. We do also have the cytoplasm and the ribosomes, which are shared, eukaryotic and prokaryotic. And then in this case, we have a cell wall in a prokaryotic cell, a capsule sometimes, a pilus, a flagellum, fimbria, and then we have the inclusions that we've mentioned briefly. So we're going to take a moment to go through each of these. So common cell morphologies and arrangements. So individual cells of particular prokaryotic organisms are typically similar in shape. So when we're talking about cell shape, we're talking about cell morphology. Although thousands of prokaryotic organisms have been identified, only a handful of cell morphologies are commonly seen microscopically. So these are some things that we've already talked about, like the coccus shape, the bacillus shape, these types of things. So even though there are so many thousands of different types of prokaryotic cells, or you can even think of bacteria, we find only these very small similar shapes, only a handful of these cell morphologies. So in addition to the cellular shape, prokaryotic cells of the same species may group together in certain distinctive arrangements depending on the plane of cell division. So let's take a look at this chart down here. Something that we are already familiar with from previous chapters, we have our different shapes here. So our common prokaryotic cell shapes, the name of them, the description, illustration, and then a nice microscopic image there. So remember, coccus shaped or cocci are round, so we see our round spherical shape there. The bacillus shape is the rod shape. You can see that there. Remember, vibrio is a rod shape that's then curved, so you can see that there. And then the coccobacillus is a shorter rod, so it's kind of round, kind of rod shaped. So these are some that we're already familiar with, these different types of shapes. And as well as our sporillum, which is the spiral shape, the spirochete, which is our long, loose, kind of telephone cord type shape as well. So these we've already taken a look at. Those are some cell morphologies. Now I just want to take a closer look at some of these cell arrangements. And some of these may already be familiar to you. So the name of them, so coccus, is a single coccus shaped, so a single round shaped cell. Then Diplococcus, which we've actually seen on some slides before, Diplococcus, di meaning two, gives us then two coccus-shaped cells, so a pair of two cocci, which then these organisms that have this type of shape are seen in these pairs. Same thing here with our tetrad. So this is the grouping of 4 cells arranged in a square. So we'll see some organisms that are arranged in a tetrad. And then this one should be familiar to you already just from readings, Streptococcus. So when we hear Streptococcus already, we can think of the coccus part. So we already know we're looking for something that's round. Now the strepto part is going to indicate its arrangement. So the strepto part is a chain, and then coccus of cocci shaped. So when you are looking for something in a microscope, or when you're trying to identify something, if you know that the name is Streptococcus, then you know that you can be looking for a chain of round-shaped bacteria. Similarly, here we have Staphylococcus. So we remember that coccus is round, and then staphylo is for cluster. So if we're looking for something that is a Staphylococcus bacteria, then we know that we're looking for a cluster of these round-shaped bacteria. Then we have our cell arrangements. We have our bacillus, which is our single rod, and then we have streptobacillus. So again, strepto meaning chain, and then bacillus meaning rod-shaped. So this would be our chain of rods. So I think this is important to go over as you're looking through microscopic images or images of through a microscope, so that you can identify a bacteria in that way. If you see these chains, then you know it's a strepto. If you see circles, it's coccus. If you see rods, it's bacillus. So those types of naming not only are helpful for us when we're looking at things, and for microbiologists or pathologists when they're looking at things, it's just been a nice handy way of naming things to make it very clear what it is that they're looking at. All right, so for most prokaryotic cells, morphology is maintained by the cell wall in combination with the cytoskeletal element. So remember, a cytoskeleton is kind of the name implies, it's a skeleton of fibers that are composing the inside. It's kind of keeping its shape that way. So we have our cytoskeleton, which is helping to keep the shape, but then also the cell wall, which is helping to keep the shape. The cell wall is a structure found in most prokaryotes and some eukaryotes. It envelops the cell membrane, protecting the cell from changes in osmotic pressure. So we're going to briefly go through osmotic pressure. This should have been covered in some of the other biology classes, but the reason it is covered here is because it is related to microorganisms, and that bacteria that have a cell wall, or prokaryotes that have the cell wall, are affected a little bit differently than, say, a eukaryotic organism that does not have, or a eukaryotic cell that does not have that cell wall. So recall that osmotic pressure occurs because the differences in the concentration of solutes on opposing sides of a semi-permeable membrane. So what we're talking about here is that plasma membrane that surrounds the cell. In both prokaryotic organisms and eukaryotic organisms, we have cells that are surrounded by that plasma membrane. And recall that water is able to pass through the semi-permeable membrane. But solutes, so larger things, are not able to get through. So we can think of proteins, for example. Proteins are quite large, so they're not able to just flow right through the membrane. So when the concentration of solutes is greater on one side of the membrane, water diffuses across the membrane from the side with the lower concentration of solutes, which would then mean it has more water, to the side with the higher concentration of solutes, the side that has less water, until the concentrations on both sides become equal. So this diffusion of water is called osmosis. Okay, and it can cause extreme osmotic pressure on a cell when its external environment changes. So recall that osmosis works where if you have one side that has few solutes and the other side has lots and lots of solutes, the water is going to go in the direction of the higher amount of solutes. I think of it as it's trying to go over and make both sides the same. It's trying to dilute the side that has the more solutes to make it seem like it's equal on both sides. So if more water goes to the side that has more solutes, then it will be more diluted and it will equal the side that has less solutes. Okay, so it's going to go to the side of more solutes. The external environment of a cell can be described as an isotonic, hypertonic, or hypotonic medium. So review, an isotonic medium is where the solute concentrations inside and outside of the cell are about equal. So there's no net movement of water. Water's not moving primarily in one direction. It still moves back and forth, but not mostly in one direction. In a hypertonic medium, the solute concentration outside of the cell exceeds that of inside of the cell. So hyper means more than. So hypertonic means that the outside of the cell has more solutes than the inside. In a hypotonic, hypo meaning less than, medium, the solute concentration inside the cell exceeds that of outside the cell. So the solution that it's in is lower, hypotonic, less solutes. Okay? So when we use those terms, hypertonic and hypotonic, we're talking about what the cell is in. Hyper meaning more, so more solutes in that, and hypo meaning less than, less solutes in that solution. So then, the solute concentration inside the cell exceeding that of outside the cell, water will move by osmosis into the cell. So when water moves by osmosis into the cell, what we can see is a swelling of the cell and potentially a lyse or a bursting of the cell. So if there are more solutes inside of the cell, remember water is going to move where there are more solutes, so it will go to inside of the cell and it can swell up and swell up and swell up, and if it keeps bringing water in, it can burst or lyse. So the degree to which a particular cell is able to withstand changes in osmotic pressure is called tonicity. So cells that have a cell wall are better able to withstand these subtle changes in osmotic pressure and maintain their shape. So this is one of the important things about prokaryotic cells, is that the cell wall is going to really help so that when the pressure changes, the cell is not going to burst, hopefully. It has a little bit more leeway in that it won't burst right away, whereas a eukaryotic cell might. And it won't crenate and die because it has a lack of water. So in hypertonic environments, cells that lack a cell wall can become dehydrated, causing crenation. So like a eukaryotic cell, if you put a blood cell, for example, a eukaryotic blood cell, in a hypertonic solution, all of the water is going to move out of that blood cell, and you can actually see the blood cell crenate. So it becomes very spiky. It's losing all of its water, shriveling up of the cell. So the membrane, the plasma membrane, is going to contract and appear scalloped or notched or kind of spiky. By contrast, so cells that possess a cell wall undergo what's called as plasmolysis. So plasmolysis is not crenation. So when we're talking about crenation, we're talking about organisms that do not have a cell wall around their cells. Plasmolysis is what occurs when there is a cell wall. In plasmolysis, the plasma membrane contracts and detaches from the cell wall, and there is a decrease in the interior volume, but the cell wall stays intact. So it allows that cell to maintain some shape and integrity for a period of time. So it allows that cell to then maintain that space. It's kind of like a space holder. So it'll crenate, but since it has that cell wall, it's going to hold that spot in the hopes that soon it will get more water and it will fill back up again. So likewise, cells that lack a cell wall are more prone to lysis in hypotonic environments. So if there is no cell wall, then we see that it's more prone to lysis. The cell wall, in the case of hypotonic solutions, allows it to maintain its shape and integrity. Let's get down to the words here. Oh, I guess it's the end. Maintain shape and integrity there we go, before a longer time before lysing. So in that same way, if we have a hypotonic solution that it's sitting in, then water is going to move inside the cell to try to dilute the contents of the cell because there are more solutes inside of the cell. When that happens, if it, for example, is a eukaryotic cell, if it's a blood cell, it will swell up and then it will burst because there's nothing helping it out there. It'll just burst because it falls apart. Too much water inside of it, like a balloon. However, in a prokaryotic cell, when it has that cell wall, it's kind of a rigid outer layer that is going to help it to withstand that for longer. So it's not going to burst as quickly. So here is an illustration of these different things. Our isotonic solution, water's moving in and out of the cell, but there's no net movement. In a hypertonic solution, water is going to move out of the cell. You'll see in eukaryotic organisms that there's crenation. In prokaryotic organisms, it's plasmolysis. And in C, our hypotonic solution, water is going to move into the cell. And then in the case of a eukaryotic cell, we'll see lysis or bursting. But we don't see that as much in those that have a cell wall. All right, here's our cell wall example. So here's our isotonic solution moving in and out. Things pretty much stay the same. Here's our hypertonic solution. So water's moving out, but you can see this is our cell wall here that's protecting the cell. And we have plasmolysis, where it shrinks down, but it stays in there. And then we have a hypotonic solution. So water's moving into the cell. It's going to swell, and you can see it even pushes on these cell walls, but the cell walls are helping to prevent it from swelling so much and then lysing. All right. So let's talk about the different parts of our prokaryotic cell now. And so some of these are different from the eukaryotic cells. You should review the material for the eukaryotic cells on your own and look back to maybe some notes that you recall from other courses. And we're going to kind of dig into the prokaryotic cell here. So first we'll talk about the nucleoid. So the nucleoid, remember, in a prokaryotic cell we don't have a true nucleus. We have what's called a nucleoid. So here all cellular life has DNA, a DNA genome organized into one or more chromosomes. So prokaryotic chromosomes are typically circular, haploid, meaning unpaired. If you recall in eukaryotic organisms, we have paired chromosomes. In a prokaryotic organism, they're typically haploid, which is unpaired, and not bound by a complex nuclear membrane. So we don't see that nuclear membrane. That's also a plasma membrane inside of the cell. So prokaryotic DNA and DNA-associated proteins are concentrated within this nucleoid region of the cell. So this is just a general area, and this can change, but what this area is just kind of a way the cell can kind of keep these things together, kind of keep them all in the same sort of area, which can change and move, but mostly it stays in the same area. So remember, the nucleoid region is prokaryotic DNA and DNA-associated proteins, which the prokaryotic DNA interacts with, which is the nucleoid-associated proteins. So the NAPs, or the nucleoid-associated proteins, you can see it's associated with the nucleoid, and their proteins, assist in the organization and packaging of the chromosome. So we see this also in eukaryotic organisms or eukaryotic cells, and this is the way a prokaryotic organism does it with these NAPs. So in bacteria, NAPs function similar to histones, which are the DNA organizing proteins in eukaryotic cells. That's what I was just talking about. So in archaea, the nucleoid is organized by either NAPs or histone-like DNA organizing proteins. So in archaea, we can see either of these things. Bacteria has the nucleoid-associated proteins and the DNA in the nucleoid region. In archaea, we can see either the NAPs, the nucleoid-associated proteins, or histone-like DNA organizing proteins along with the DNA. And you can see that region there, the nucleoid region. Also important, if we're talking about the nucleoid, we're talking about DNA. That's where the DNA is kind of not housed, but it's where it hangs out. In prokaryotic cells, you may also contain what's called extrachromosomal DNA. So extra meaning outside of, chromosomal, so outside of that main chromosome. So we'll see more DNA, or DNA that is not part of the chromosome. This extrachromosomal DNA is found in plasmids. So we oftentimes see plasmids in bacteria or other prokaryotic cells. And these are small, circular, double-stranded DNA molecules. So basically it's like a little loop of DNA that's hanging out all by itself outside of the nucleoid region. Cells that have plasmids often have hundreds of them within a single cell. So it'll have all of these little tiny rings within a single cell. Plasmids are more commonly found in bacteria. However, plasmids have been found in archaea and eukaryotic organisms. Plasmids often carry genes that confer advantageous traits, such as antibiotic resistance. So they're very important to the survival of the organism. So we're going to talk about that a lot more in another chapter. But generally, we see plasmids mostly in bacteria. When we see them in bacteria, most times they have antibiotic resistance associated with them. So they're very important plasmids, and that actually allows them to be manipulated very easily by researchers as well, but we can talk about that in that chapter. So ribosomes, all cellular life synthesizes proteins, and organisms in all three domains of life possess ribosomes. So remember, that's one of those four things that all cells share, eukaryotic and prokaryotic. So ribosomes, remember, are structures that are responsible for protein synthesis. So they're the things that make the proteins. However, the ribosomes in each of the three domains are structurally different. So ribosomes themselves are constructed from proteins, along with ribosomal RNA, if you remember rRNA. Prokaryotic ribosomes are found in the cytoplasm. They are called 70S ribosomes, because they have a size of 70S, whereas eukaryotic cytoplasmic ribosomes have a size of 80S. And then a note here that the S stands for the Svedberg unit, which is the measure of sedimentation in an ultracentrifuge, which is based on size, shape, and surface qualities of the structure being analyzed. So the S is just based on the person that was able to take ribosomes, put them in a centrifuge, an ultracentrifuge, and measure how much sediment there is. That part's not important for you to recall. You should recall, or you should know, that prokaryotic ribosomes are floating around in the cytoplasm, and that they're called 70S ribosomes, versus eukaryotics, which are called 80S ribosomes. So although they are the same size, bacterial and archaeal ribosomes have different proteins and rRNA molecules. So although they are similar in size, different proteins, different rRNA molecules, and the archaeal versions are more similar to the eukaryotic ribosomes rather than those found in bacteria. So remember, when we looked at our phylogenetic trees, we also saw that archaea are a little bit more similar to eukarya versus being similar to bacteria. So we'll see that archaea is similar in some ways to the bacteria that we're going through, and then similar in other ways to the eukarya. All right. So up here, we can see actually an illustration of that. So our prokaryotic ribosome, which is 70S, is composed of two subunits. If you remember, eukaryotes also have a ribosome composed of two subunits. So the 30S subunit, that's this guy here, the smaller one, they colored it blue, and the 50S subunit, which is the large one, which is this guy here, colored in kind of this peachish color. So both of these are composed of protein and rRNA components. And then we have the complete ribosome, which is the 70S, which of course is 50, the 50S plus the small unit, which is the 30S. That doesn't add up to 70, but that is nonetheless what it's called, is the 70S complete ribosome. All right, so let's move on to our inclusions. So our single-celled organisms live in unstable environments. Lots of times we've talked about in previous chapters how they're extremophiles, how they're bacteria and archaea that are able to live in very extreme areas. So some prokaryotic cells have the ability to store excess nutrients within the cytoplasmic structures called inclusions. So we have inclusions inside these prokaryotic cells that are going to store the nutrients. So if they're in a situation, the bacteria, for example, are in a situation where they're surrounded by lots and lots of nutrients, they're going to store some of those nutrients that they don't need to function immediately in inclusions. So storing nutrients in a polymerized form is advantageous for the organism because it reduces the buildup of osmotic pressure that occurs as a cell accumulates solutes. So if you think back to the osmotic pressure, if the cell had all of these nutrients, say it was in a situation where it had lots and lots and lots of nutrients available to it, and it just brought all of those things in and they were all hanging out inside the cytoplasm, then that would seem that the cytoplasm in there would have lots and lots of solutes. And then what we would see would be a rushing in of water. So if there's a rushing in of water, then we're going to have a situation where the cell is going to swell up. And even though there's a cell wall there protecting it, that can only hold for so long and only so much water. So then the buildup of osmotic pressure would perhaps lyse the cell. So instead, the bacteria have inclusions, and so then they're able to have this polymerized form where it stores these nutrients, and then it's not going to contribute to the osmotic pressure because you've got these inclusions rather than all of these solutes floating around in the cell. So various types of inclusions store glycogen and starches, so our sugars there, which contain carbon that cells can access for energy. So it's like an energy storage. Our inclusions are an energy storage area, cytoplasmic structure. Volutin granules, also called metachromatic granules, because of their staining characteristics, chromatic meaning color, they stain, are inclusions that store polymerized inorganic phosphate that can be used in metabolism and to assist in the formation of biofilms. And we're going to talk about biofilms in a little bit here. So we have inclusions that can store glycogen and starches, which contain carbon that can be used for energy. And then we have very specific ones that are called volutin granules or metachromatic granules. And they store this polymerized inorganic phosphate that can be used in metabolism and to make biofilms, which are important for the success of some bacteria, to be able to have biofilms to protect them. Microbes known to contain volutin granules include archaea, the methanosarcina, the bacterium Corynebacterium diphtheria, and the unicellular eukaryotic alga Chlamydomonas. So those are some examples of microbes that have these volutin granules that are very helpful because they're able to store this phosphate for later use. There are also sulfur granules, which is another type of inclusion, and they're found in sulfur bacteria, so the thiobacillus. And then these store the elemental sulfur, which this type of bacteria uses for metabolism. So as you can see, these are different ways that the bacteria can store things to then later use them for metabolism so that they can function in areas where they may not have nutrients. Occasionally, certain types of inclusions are surrounded by a phospholipid monolayer embedded with protein. So polyhydroxybutyrate, or PHB, can be produced by a species of Bacillus and Pseudomonas. Bacillus and Pseudomonas is an example of an inclusion that displays this type of monolayer structure. So our polyhydroxybutyrate, our PHB, can be produced by some Bacillus and some Pseudomonas, and that actually is kind of like a nucleus in a eukaryotic cell, whereas it has this phospholipid layer. However, it's not a bilayer; it's a monolayer that has some proteins embedded in it. So it's a little bit of a different type of inclusion, produced only by some species. But it's mentioned here because industrially, PHB has also been used as a source of biodegradable polymers for bioplastics. So those can be very helpful, and I think those are mentioned here because that phospholipid monolayer with the protein that's produced by these bacteria. It can be produced and then be used for this biodegradable polymer. Another way that we can use bacteria to help things to be useful. That's something that we can exploit them and use them in that way. So some prokaryotic cells have other types of inclusions that serve purposes other than nutrient storage. So all of the ones that we just went through there were about nutrient storage. So if they're in a situation where they find themselves with a lack of nutrients, they can then use those stores. Some other ones, for example, prokaryotic cells produce gas vacuoles, so accumulations of small protein-lined vesicles of gas. These gas vacuoles allow these prokaryotic cells that synthesize them to alter their buoyancy so that they can adjust to their location in the water column. So we can see that it's kind of similar to the way some water organisms are able to adjust their buoyancy when they have these air bladders in them to allow them to move up in the water or down in the water depending on how much gas is in there. So how much air, how much gas is in there. So we can see some prokaryotic cells have these gas vacuoles that are protein-lined vesicles that kind of hold gas in and allows them to adjust their location in water. Magnetotactic bacteria, such as the Magnetospirillum magnetotacticum, contain magnetosomes. So these are inclusions of magnetic iron oxide or iron sulfide surrounded by a lipid layer. So again, we have this kind of layer, this inclusion that keeps it separate from other things, a lipid layer that then has magnetic iron oxide in it or iron sulfides. And these allow cells to align along a magnetic field. So it also allows, it aids in their movement. So similar to these gas vacuoles that can move, allows them to move, we have these magnetotactic bacteria that have this iron oxide that allows them to move along a magnetic field. Cyanobacteria, such as Anabaena cylindrica, and bacteria such as the Halothiobacillus neopolitanus produce carboxysome inclusions. So carboxysomes are composed of outer shells of thousands of protein subunits. So their interior is filled with this ribulose 1,5-bisphosphate carboxylase / oxygenase, or rubisco. It's most commonly called rubisco, so you can remember that rather than the really long name that I just said there. And carbonic anhydrase. So our carboxysome, you don't have to remember those different bacteria. You can remember that cyanobacteria produce carboxysome inclusions, and those carboxysome inclusions contain rubisco and carbonic anhydrase. And both of these compounds are used for carbon metabolism. Okay, so more metabolism there. Some prokaryotic cells also possess carboxysomes that sequester. Remember, it's these, we're talking about inclusion, so we're talking about sequestering these enzymes in one location, functionally related enzymes in some location. So another way that they can kind of keep things from just floating around the whole organism, they have carboxysomes that are going to take enzymes and keep them all in one spot. So these structures are considered proto-organelles because they compartmentalize important compounds or chemical reactions like eukaryotic cells or eukaryotic organelles. So typically, that's why we say in general we don't see these compartmentalized organelles like we see in eukaryotic organisms or eukaryotic cells. However, this is one example of that. So we're talking about inclusions, and one of the types of inclusions they see are these carboxysomes that are going to keep enzymes in a certain area so that they can be used at a different time. All right, so you should know all of these different types of inclusions, what an inclusion is, why it's helpful, the metachromatic granules, some of the other inclusions, the volutin granules, the PHB. Remember some of these down here, the magnetotactic bacteria, the carboxysomes, And now we're going to move into our endospores. So bacterial cells are generally observed as vegetative cells, but some genera of bacteria have the ability to form endospores. So the endospores are structures that essentially protect the bacterial genome in a dormant state when environmental conditions are unfavorable. So endospores are actually the way that a cell, when it's in an unfavorable environment, are going to kind of hunker down and kind of collect themselves and kind of go into this dormant state. So endospores allow some bacteria to survive for long periods of time without food or water. So you can actually think of it as kind of a hibernation like we see in some animals, where when the bacteria finds itself in a state where there's not many nutrients, it's unfavorable, it can kind of go into this hibernation state, and everything kind of slows down, so it doesn't need as much food, it doesn't need as much water, and it can kind of stay in this state until it's then introduced to an environment that's more beneficial for it, has food, has nutrients, water, and then it'll kind of open itself back up and start going through its metabolic processes again. So they can survive long periods without food or water, as well as exposure to chemicals. So we're talking about different types of unfavorable environments that they might find themselves in. So chemicals, extreme temperatures, and even radiation. So they can kind of go into this shutdown mode. Also, it mentions here, so endospores, not to be confused with the reproductive spores formed by fungi. So we are going to talk about fungi in a little bit more detail. The spores that are formed by fungi are reproductive spores. So those are the things that are sent out there kind of like a seed to make more fungi. And endospores are then this kind of hibernation state of these bacterial cells. So again, we generally see them as their vegetative cells, but then they can then go into this endospore type of cell. And then here is a chart that's talking about the vegetative cells, which is how we normally see bacteria kind of moving around in a broth or on a plate, for example, versus endospores. So the vegetative cells, kind of in their regularly living state, are sensitive to extreme temperatures and radiation, depending on the bacteria, of course. So if it's an extremophile, that's a type of bacteria that is used to living in extreme cold or extreme hot. But in general, bacteria like a bacillus that we would see in a laboratory or in a human body is very sensitive to an extreme temperature or radiation. Whereas when it is in its endospore forming stage, it's resistant to extreme temperatures and radiation. Vegetative cells are gram-positive. Those that form endospores are gram-positive cells. And then the endospores do not absorb the gram stain. So they only absorb special endospore stains. And we're actually going to see that in the future, but we've already talked about how there's a specific endospore stain that turns them green. In the vegetative cells, we see normal water content and enzymatic activity because it's in its typical living state. In endospores, they're dehydrated. They don't have any metabolic activity. And in the vegetative cells, they're capable of active growth and metabolism. And in the endospore state, they're dormant, so they don't have any growth or metabolic activity. So the process by which these vegetative cells, these living organisms, transform into endospores is called sporulation. And it generally begins when nutrients become depleted. So it finds itself in this depleted nutrient environment. Or the environmental conditions become otherwise unfavorable. So radiation or extreme temperatures and such. The process begins with the formation of a septum in the vegetative bacterial cell. The septum divides the cell asymmetrically, so rather than symmetrically, which would be perfect halves, asymmetrically is not symmetrically, separating a DNA 4 spore from the mother cell. So meaning that there are two different things that are formed. There's a DNA 4 spore and then the mother cell. So the 4 spore, which will form the core of the endospore, is basically a copy of the cell's chromosomes. So this spore. Remember, the septum is going to divide the cell to where it has this mother cell, and now we're talking about the spore, which is basically just a copy of the cell's chromosomes. And then it's separated from the mother cell by a second membrane. So it has its own little membrane that's protecting a copy of the DNA. A cortex then gradually forms around that spore by laying down layers of calcium and dipicolinic acid between the membranes. So we form this membrane, so there's the second membrane inside there. We've got the spore forming with its chromosomes in there, and then we have this cortex that forms around the spore with the calcium and the dipicolinic acid between the membranes. So a protein spore coat is then going to form. So we have, again, the cortex that forms, and then this protein spore coat that forms around the cortex while the DNA of the mother cell disintegrates. So it's made a copy of the DNA, it's protecting that DNA in that spore that has these layers around it, so the cortex on it and then the protein spore coat on it, and then the DNA in the mother cell is going to disintegrate. Then there is further maturation of the endospore that then ends with the formation of an outermost exosporium. So then we have this. So we have these layers that are making kind of this this capsule. Well, I don't want to use capsule because that's different, but this really strong outer layer, several outer layers that are going to protect that DNA. Then the endospore is released upon disintegration of the mother cell, and that completes sporulation. So we kind of make a copy of the DNA, and then that DNA goes into what's called a spore, which is surrounded by a membrane, and then the mother cell is kind of what's left, and then this membrane has a cortex form around it with calcium and dipicolinic acid, and then a protein spore coat forms around that cortex, and then there's the formation of the outermost exosporium, and then that endospore is released once that mother cell is degraded. And then that completes sporulation. So here is an illustration of that. So A is sporulation begins following asymmetric cell division. So here we have our DNA replicates, so we make a copy of the DNA. Then we have our membrane, you can see, forming around that copy of DNA. We have our spore forms, additional membranes. So you can really see that it's kind of turning into this own little membrane-bound sac here, membrane-bound sphere. Then we have our protective cortex that forms around the spore, so you can see the kind of yellow that's been added in this area. Then we have our protein coat that forms around that cortex. You can see it's much thicker now. And then we have our spore release. So this is then going to be disintegrated, all of this stuff out here, and then we have our spore. Then you can see down here, you can see in B is an electron micrograph of a spore here, of an endospore that has been formed. And then in C, we have the cells that are undergoing sporulation. You can see the green right there is an endospore that's forming. These are endospores, all of the green. And then here you can see it mentions this is the vegetative cell, so just the typical living everyday bacteria cell, and then the green one is the endospore. So endospores of certain species have been shown to persist in a dormant state for extended periods of time, so up to thousands of years. So the endospore is incredibly strong. However, when living conditions improve, the endospores undergo what's called germination. So germination is this re-entering of a vegetative state. So vegetative meaning it's going to go back into its everyday, using up of nutrients, metabolizing things, going through growth. After germination, the cell becomes metabolically active again and is able to carry out all of its normal functions, including growth and cell division. So not all bacteria have the ability to form endospores. So there are only certain types of bacteria that can form endospores, but there are clinically significant endospore-forming bacteria. So again, bacteria that can form endospores is gram-positive. So remember, the endospore itself is not gram-positive. The vegetative state of that type of bacteria is gram-positive, and they are Bacillus and Clostridium genus. The genus is Bacillus or Clostridium. Those are the types of cells that are able to form endospores. So some of the important ones are anthrax, the bacteria that causes anthrax, the Bacillus anthracis, the causative agent of anthrax. It produces endospores that are capable of surviving for decades. So that's why we had that kind of bioterrorism thing that people were talking about for a while with anthrax, because they have the spores, and then the spores can be released and they can survive for decades. C. botulinum causes botulism. Oh, I skipped a couple here. C. difficile, which is the pseudomembranous colitis. C. perfringens, which causes gangrene or gangrene. And then pathogens are particularly, these types of pathogens are difficult to combat because the endospores are so hard to kill. So if you can imagine, if they last thousands and thousands of years, endospores are very, very strong. And so some of these pathogens are incredibly difficult to get rid of. All right. So then I'll just briefly mention the plasma membrane. So you should be familiar with the plasma membrane. You should know the plasma membrane from eukaryotic cells. So structures that enclose the cytoplasm and internal structures of the cell are known collectively as the cell envelope. So when we're talking about the structures that are enclosing these things, they're the cell envelope. So in prokaryotic cells, the structures of the cell envelope vary depending on the type of cell and the organism. Most of them have a cell wall, and the makeup of the cell wall varies. We have already talked about that and seen that with gram-positive versus gram-negative. All cells, prokaryotic and eukaryotic, have this plasma membrane, so also called the cytoplasmic membrane or cell membrane. So I'm not going to go through the plasma membrane here. You should be familiar with it, the fluid mosaic model, what it's made up of, the different types of proteins, glycoproteins, lipids, how it works. Okay, so the archaeal membranes are fundamentally different from bacteria and eukaryotic membranes in a few ways. So first, the archaeal membrane phospholipids are formed with ether linkages. So in contrast to the ester linkages found in bacteria or eukaryotic cell membranes. So the phospholipids, I can scroll back up here, maybe down here. So up here we have our phospholipids. Remember, our cell membrane made up of phospholipids. We have our phospholipid bilayer. So the phospholipids in our bacterial cells and the eukaryotic cells are formed with ester linkages, and in archaea they're formed with ether linkages. Second, our archaeal phospholipids have branched chains, whereas those of the bacteria and eukaryotic cells are straight chained. So again, if we look up here, we see our phospholipids. You can see the kind of tails on there, the chains on our phospholipid. So as you can see, they're just straight chains hanging down there. However, what we see in archaea is that they have branched chains. Finally, although some archaeal membranes can be formed of bilayers, like those found in bacteria and eukaryotes, other archaeal plasma membranes are lipid monolayers. All right, so those are the three main differences between our bacteria and eukarya membranes, plasma membranes, and archaea. So those three things, archaea membranes, their phospholipids are formed with ether linkages instead of ester linkages. Second, the phospholipids have branched chains in archaea versus the straight chains in bacteria and eukaryotic. And then the archaea membranes can be bilayers, just like bacteria or eukaryotic cells, or they can be monolayers. So proteins on the cell surface are important for a variety of functions, including the cell-to-cell communication, just like we see in eukaryotic and also bacteria, and sensing environmental conditions and pathogenic virulence factors. So membrane proteins and phospholipids may have carbohydrates on them, which are glycoproteins, glycolipids. This is kind of general things. Our glycoprotein and glycolipid complexes extend out from the surface of the cell and allows the cell to interact with the external environment, just like in eukaryotic cells. Glycoproteins and glycolipids in the plasma membrane can vary depending on, or can vary considerably in chemical composition among the archaea, bacteria, and eukaryotes. So they can look at those different glycoproteins and look at the different glycolipids, and they can determine the chemical composition. And when they determine the chemical composition, they can also characterize them based on the archaea, bacteria, and eukaryotes. However, you can see all three of them have those glycoproteins and glycolipids in them. Plasma membranes from different cell types also contain unique phospholipids, which contain fatty acids. Phospholipid-derived fatty acid analysis, or PLFA, profiles can be used to identify the unique types of cells based on the differences in these fatty acids. So archaea, bacteria, and eukaryotes each have their unique PFLA profile. So they have slightly different phospholipid-derived fatty acids on there. So just general differences there. You don't have to know specifically those differences. You don't have to know what's different about different glycoproteins or glycolipids for archaea, bacteria, eukaryotes. Just know that they do have differences and they can use them to identify them. And then the same thing with the PFLA profile. So you don't have to know what those differences are, just know that archaea, bacteria, and eukaryotic cells have differences in those phospholipids. All right, so membrane transport mechanisms, just go over it really quick. Again, I have other things posted that can go through this again. You should be familiar with this already. One of the most important functions of the plasma membrane is to control the transport of molecules into and out of the cell. So they need to be within this certain range. Cells like to have everything be balanced. So there are different modes of transport across the plasma membrane. We already talked about things moving from a higher concentration to a lower concentration with the concentration gradient. That's called simple diffusion. Things like that happen with small molecules. I'm not going to go through all these different types of transport. You can do that on the lecture that I've already posted. It shows simple diffusion, active transport, facilitated diffusion. It talks about the different types of diffusion there and osmosis. So you can listen to that lecture. Here I will talk about our photosynthetic membrane structures. So some prokaryotic cells, namely cyanobacteria and photosynthetic bacteria, have membrane structures that enable them to perform photosynthesis. So that's different than some eukaryotic cells. So we see, of course, the photosynthetic aspect of plants, not human cells, but plant cells, which is a eukaryotic cell. But we also see photosynthetic bacteria and cyanobacteria that can perform photosynthesis. So these structures consist of an infolding of the plasma membrane that then encloses photosynthetic pigments, such as green chlorophylls and bacteria chlorophylls. So similar to a plant cell, it has chlorophylls in it. So you can see an infolding, folding in of the plasma membrane that will then enclose these photosynthetic pigments, so chlorophylls and bacterial chlorophylls. In cyanobacteria, these membrane structures are called thylakoids. In photosynthetic bacteria, they're called chromatophores, lamellae, or chlorosomes. Okay, so you should know those things. You should know what a lamellae or a chlorosome or a chromatophore is, or a thylakoid. So know that in photosynthetic bacteria, they are the chromatophores, lamellae, or chlorosomes. In cyanobacteria, they're called thylakoids. All right, so moving on to the cell wall. The primary function of the cell wall, we've already talked about it, is to protect the cell from harsh conditions in the outside environment. So when it's present, there are notable similarities and differences among the cell walls of archaea, bacteria, and eukaryotes. Okay, so remember, in eukaryotes, we see the cell wall in plant cells, not in human cells. And then we see it in archaea and bacteria. We've already talked about it a bit. So the major component of a bacteria, or a bacterial cell wall, is peptidoglycan. And so peptidoglycan, again, we've already seen that with the gram stain. With a gram stain, remember, gram-positive has more peptidoglycan, it's thicker layers, versus a gram-negative, which is a thinner layer, so it doesn't hold that purple stain in as much. Is only found in bacteria. So when we talk about peptidoglycan, we're talking about a bacteria cell wall because it's only found in bacteria. So structurally, peptidoglycan resembles a layer of meshwork or fabric, and each layer is composed of long chains of alternating molecules of NAG and NAM. Okay, N-acetylglucosamine is NAG, N-acetylmuramic acid is NAM. So the structure of the long chains has significant two-dimensional tensile strength due to the formation of these peptide bridges that connect the NAG and NAM within each layer. Okay, so we have the NAG and the NAM, which are different molecules, and then the structure of these long chains has this really strong strength, so this tensile strength because of the bridges between these two molecules, between the NAG and the NAM within each peptidoglycan layer. So in gram-positive, remember, there are many layers, and then in gram-negative, there are fewer layers. So in gram-negative bacteria, tetrapeptide chains extending from each NAM unit are directly cross-linked. Whereas in gram-positive bacteria, these tetrapeptide chains are linked by pentaglycine cross bridges. Okay, so what we see in gram-negative bacteria, we have tetrapeptide chains. Tetra is 4 peptide chains that extend from each NAM cross-linked, versus in gram-positive bacteria, we see these tetrapeptide chains are linked by pentaglycine cross bridges. Okay, so peptidoglycan subunits are made inside of the bacterial cell and then exported and assembled in layers, giving the cell its shape. So within the bacteria, it produces and makes these subunits, the peptidoglycan subunits, and then it exports them or brings them outside of the cell to go ahead and create that cell wall. And we can see here, I'll scroll down just a little bit more, so you can see our gram-positive here cell and our gram-negative cell. Down here it mentions peptidoglycan is composed of polymers of alternating NAM and NAG subunits, which are cross-linked by peptide bridges linking the NAM subunits from various glycan chains. So this provides it with tensile strength in 2 dimensions. So you can see here's the NAG, the N-acetylglucosamine, and then the NAM, N-acetylmuramic acid. And you can see, so the NAM are these orange guys, and then the NAG are the blue ones. And you can see here in our gram-positive, the pentapeptide here, oh yeah, penta, sorry, not 4 or 5. 1, 2, 3, 4, 5, penta, like, yes. And then the tetrapeptide, 1, 2, 3, 4, tetra. So our pentapeptide and tetrapeptide connecting these pieces here, the NAM and the NAG. And then here in gram-negative, we have the NAG, again, the blue ones, and then the NAM, the orange ones. And then we have these direct links here. So you can see the direct links that are connecting them. All right. So since peptidoglycan is unique to bacteria, many antibiotic drugs are designed to interfere with peptidoglycan synthesis so that it can weaken the cell wall and then making bacterial cells more susceptible to the effects of osmotic pressure. So again, remember, the cell wall is something that helps protect the bacterial cell from osmotic pressure, from the exploding of the cell, the lysing of the cell, or the crenation of the cell. Instead, we have the plasmolysis of the cell, and then the cell wall protects it from bursting. So if we are able to make antibiotic drugs, a lot of them are made to interfere with this production of the cell wall. And then if we interfere with the production of the cell wall, it makes the bacteria more susceptible, and then it can go through that process of lysing. In addition, certain cells of the human immune system are able to recognize bacterial pathogens by detecting peptidoglycan on the surface of a bacterial cell. These cells then engulf and destroy the bacterial cell using enzymes such as lysozyme, which breaks down and digests that peptidoglycan in their cell walls. So some of the cells in our body, we have macrophages, they're called, that are kind of rooting around, white blood cells that are moving throughout our body. They can sense or pick up on the peptidoglycan, and then when they notice that there's peptidoglycan, the macrophage knows that other body cells don't contain peptidoglycan, that's a part of bacterial cell, then it will engulf that and destroy that bacterial cell. So the Gram staining protocol is used to differentiate two common types of cell wall structures. So the gram-positive cells have a cell wall consisting of many layers of peptidoglycan, about 30 to 100 nanometers in thickness. These peptidoglycan layers are commonly embedded with teichoic acids, TAs, carbohydrate chains that extend through and beyond the peptidoglycan layer. TA is thought to stabilize the peptidoglycan by increasing its rigidity. So you can imagine if there are more layers of peptidoglycan, and then we have these TAs that are these carbohydrate chains that are going to extend through them and beyond past or go past them, that they're going to be more stable and more rigid. So TA also plays a role in the ability of pathogenic gram-positive bacteria, such as Streptococcus, to bind to certain proteins on the surface of the host cells. So these carbohydrate chains, in general, carbohydrates are kind of sticky. So these carbohydrate chains, the TA, stick off of these gram-positive bacteria and allow them to bind to certain proteins in the organism that it's trying to infect in the host cell, which enhances their ability to cause infection. So if they're going to be able to stick to the cells, then they're not going to be washed away as easily. In addition to peptidoglycan and TAs, bacteria of the family Mycobacteriaceae have an external layer of waxy mycolic acids in their cell wall, as mentioned in the staining part. So we've already talked about mycolic acid and why that's important, and that's something that you should still know and remind yourself of, that acid-fast organisms have that mycolic acid in their cell walls, and that's the mycobacterium. So since acid-fast stains must be used to penetrate the mycolic acid layer for purposes of microscopy. So they are called acid-fast. What makes them acid-fast is this external layer of mycolic acid, and we see that in Mycobacterium, which is kind of easy to remember. Mycobacterium has mycolic acid. They have the same beginning there. And then those two things, Mycobacterium that has the mycolic acid, are also referred to as acid-fast, because we use the acid-fast stains in order to determine if it is a mycobacterium. All right, so you can see here the two common cell wall structure types are gram-positive cell walls, which are over here, and the gram-positive with our plasma membrane. You can see the peptidoglycan cell wall and the TA here that are going to be cross-linking those. And then you can see our gram-negative bacteria. You can see the plasma membrane in here, and then their peptidoglycan cell wall there, which is much, much thinner than the gram-positive cells. And then we can see here we have our murine lipoprotein, we have lipid A, O antigen, lipopolysaccharide, a porin, this kind of outer membrane stuff. There's even some periplasmic space in here. So you can see that the gram-negative cell walls are very complex. They have all of this going on in the gram-negative walls. And that it even mentions down here, let's see, containing 3 layers. So we have the inner membrane here, right? And then this thin layer of peptidoglycan, and an outer membrane containing lipopolysaccharide. So then we have our peptidoglycan wall and then our outer membrane. All right, and then here's a picture of our acid-fast. So it even shows our gram-positive acid-fast that has the plasma membrane and the peptidoglycan cell wall, just like if we look up here, just like our gram-positive bacteria here has the plasma membrane and the peptidoglycan cell wall. And then we have our TA here as well, and then we have our mycolic acids. So remember, the mycolic acids are what are going to make it acid fast, and that's in our mycobacterium. Alright, so gram-negative cells have a much thinner layer of peptidoglycan, so we just talked about that, and we're going to talk about it in detail here. So they have a much thinner layer of the peptidoglycan, so no more than 4 nanometers thick, than gram-positive cells, and the overall structure of their cell envelope is more complex, which we just saw. So in the gram-negative cells, we have a gel-like matrix that occupies that periplasmic space. So we just saw that, the periplasmic space between the cell wall and the plasma membrane. And then there's a second lipid bilayer called the outer membrane, and that's external to the peptidoglycan layer. So we have that kind of gel-like matrix that we saw up here. So we have our inner membrane, and then our gel-like matrix in this periplasmic space, and then we have the outer membrane. Alright, so this right here, outer membrane, is attached to the peptidoglycan by the murine lipoprotein. So this outer leaflet of the outer membrane contains the molecule lipopolysaccharide, or LPS, and that functions as an endotoxin in infections. So if it involves a gram-negative bacteria, we have this endotoxin, which can cause fever, hemorrhaging, and septic shock. So this lipopolysaccharide can be very dangerous when we have a gram-negative bacteria. So each LPS molecule is composed of lipid A, a core of polysaccharide, and an O side chain that is composed of sugar-like molecules that comprise the external face of the LPS. All right, let me scroll down here. So we have our right here, the outer membrane of our gram-negative bacterial cell. This is our LPS here, the lipopolysaccharide. So this is a toxin, an endotoxin, endo meaning outside of, so a toxin that's outside of the bacteria, composed of lipid A embedded in the outer membrane, a core of polysaccharide, and the O side chain. So here we can see our lipid A, which again is in the outer membrane rather than the inner membrane. We have our core polysaccharide, poly meaning many, and saccharide is a sugar, and then the O antigen side chain. All right, so this is our LPS, okay, is composed of these 3 things. And these 3 things then, so we have our outer membrane of our gram-negative bacteria. Okay, and we can scroll back up and take another look at that here. So our gram-positive is pretty basic. We've got our plasma membrane, peptidoglycan cell wall with the TA, kind of linking those peptidoglycan pieces together. Then we have our gram-negative bacteria. We have our inner membrane, the gel-like periplasmic space, and then we have the outer membrane. The outer membrane, or then our, skipped our peptidoglycan wall, very thin one, remember, 4 nanometers. Then the outer membrane. And then here in the outer membrane, we have, they kind of showed in here, our lipoprotein here, and then we have our lipid A, and then our O antigen, so our lipopolysaccharide. All right. So the composition of this O side chain varies between different species and strains of bacteria. So parts of the O side chain called antigens can be detected using serological or immunological tests to identify specific pathogenic strains like E. coli 0157H7, which is a deadly strain of bacteria that causes bloody diarrhea and then kidney failure. So on this LPS, parts of this side chain, as is mentioned here, are an antigen, and so then we can identify the antigens using antibodies in serological tests or immunological tests. So that is going to summarize, I'll just scroll up one more time, this is going to summarize our bacteria cell wall, and we're going to move into the archaea cell wall. So we have our gram-positive bacteria, and then our gram-negative bacteria cell wall. You should know the different parts, how gram-positive have the plasma membrane, and then this very thick peptidoglycan wall. And then you should know the cross-linkaging here with the pentapeptide on our gram-positive and the tetrapeptide versus gram-negative. In their peptidoglycan, They have the NAG and the NAM, but then there's a direct link between the two. And then also you should know in gram-negative bacteria, there's this inner membrane, there's this periplasmic space, this kind of like gel area here. Then we also have a thin peptidoglycan wall, and then we have another outer membrane that has the LPS in it. And that those LPS molecules actually have these three parts to them. The lipid A, that's the part that is inside of that outer membrane, embedded in the outer membrane. Then it has the core, which is a polysaccharide, and then it has that O side chain that has antigens on it. It depends on the different species and the different strains, but parts of that O side chain can have antigens that can be detected in immunotests. All right. So archaeal cell wall structure differs from that of bacteria in several ways. So first, the archaeal cell walls do not contain peptidoglycan. Remember, peptidoglycan was very specific to bacteria. Instead, they contain something that's similar to peptidoglycan, but it's pseudo-peptidoglycan, and we talked about that in Chapter 1. So you should recall that bacterial cell walls have peptidoglycan, archaeal walls have pseudo-peptidoglycan. Let's see, in this case, we have the NAM that is in peptidoglycan would be replaced in pseudopeptidoglycan with a different subunit. So other archaea may have a layer of glycoproteins or polysaccharides that serve as the cell wall instead of pseudopeptidoglycan. So archaea are kind of a mixed bag. Some have this pseudopeptidoglycan that doesn't have the NAM. That piece has been changed out depending on the archaea. Some have glycoproteins, some have polysaccharides. And then last, as in the case with some bacterial species, there are a few archaea that appear to lack cell walls entirely. So that's why we say, in general, bacteria have cell walls, archaea have cell walls, but as they're looking through them, some don't, so there are always exceptions to that rule. All right, so now we're going to talk about glycalices and S layers. Glycalices, or glycolax, singular. So most prokaryotic cells have cell walls. Some may also have additional cell envelope structures that are exterior to the cell wall, such as glycalices and S layers. So glycalices is plural for a glycocalyx. So a glycocalyx is a sugar coat, of which there are two important types. So there are capsules and there are slime layers. So a glycocalyx, remember, think sugar, kind of sticky outer layer. So a glycocalyx, there are capsules and slime layers. A capsule is an organized layer located out of the cell wall and is usually composed of polysaccharides or proteins. Polysaccharide meaning many sugars, polysaccharides or proteins. And then a slime layer is less tightly organized layer. So the first one, the capsule, is very organized. It's kind of a really tight layer. You can imagine a capsule, something a little bit more solid of polysaccharides and proteins. And then the slime layer is less tightly organized. You can kind of imagine slime being loose, and it's only loosely attached to the cell wall. It can be more easily washed off. So slime layers may be composed of polysaccharides, glycoproteins, or glycolipids. Okay, so it depends again on the organism. So a capsule is more solid. It can't just be washed away. A slime layer is less loosely organized, and it can actually be washed off pretty easily. Glycocalyces allow the cells to adhere to surfaces. So we're talking about a sugar coat. If you can imagine, when you spill something that had sugar in it, it's very sticky. It starts to dry up, it's very sticky. So the glycocalyces allow the cells to adhere to other surfaces. It makes them sticky. That aids in the formation of biofilms. Biofilm is a colony of microbes that form on the layers, or in layers on surfaces. And in nature, most microbes live in mixed communities within biofilms. So a biofilm, as just mentioned here, is kind of a mixed community. There are a lot of different types of bacteria that all kind of stick together using their glycocalyx layer. And then it protects them. It's kind of, if there are more of them, then they're protected. They're a little bit more stable. So because of the biofilm, it affords them some level of protection. So biofilms generally hold water like a sponge, which prevents desiccation, so a drying out. So if they create a biofilm with even other types of bacteria, then they're able to hold water, and then it prevents all of the bacteria from desiccation or from drying out. They also protect cells from predation and hinder the action of antibiotics and disinfectants. So if something comes along to try to get rid of them, if there are antibiotics introduced, it can be such a huge layer, such a big film, that even with antibiotics, it can just kill like the outer layer, the outside part of it, and then it will still hold, the inside part will still hold true, and those organisms will keep living and then multiplying and keep creating a bigger biofilm. Same thing with disinfectants. So if there's a biofilm on a surface, disinfectant can be sprayed on there and wiped on there, and then it will act in the same way, where the kind of external ones, the ones that are on the outside area, may die because of the disinfectant or the antibiotic, but then the ones in the middle will continue living. It's a way to protect the colony. So all of these properties are advantageous to the microbes living in a biofilm, but they present challenges in a clinical setting. So where our goal is to get rid of these, let's see, it's going to be very, very difficult to get rid of them. So I don't know where that sentence went, but oh, there it is. Where the goal is often to eliminate microbes. All right, so the ability to produce a capsule can contribute to a microbe's pathogenicity, so the ability for it to cause disease. So because this capsule, remember the capsule was the really strong one, since the capsule can make it more difficult for phagocytic cells, like white blood cells, like the macrophage that I talked about, that engulfs and kills microorganisms. It makes it more difficult for that macrophage to get in past that capsule to get to the cell wall and break down the cell wall so that the cell will lyse. Streptococcus pneumonia, for example, produces a capsule that is well known to aid in this bacterium's pathogenicity. So talked about that in staining microscopic specimens, but capsules are difficult to stain for microscopy, and negative staining techniques are typically used. So again, we have these glycocalyces, and we'll talk about the S layer in just a moment, but a glycocalyx is that sticky sugar coat. There are two different types: the capsule, which is stronger, more well organized of polysaccharides or proteins, and then the slime layer, which is less tightly organized, made up of polysaccharides, glycoproteins, or glycolipids. Both of them are going to help with making biofilms. All right, so then we have an S layer. An S layer is another type of cell envelope structure. Remember, we're talking about the cell envelope is all the stuff outside of the cytoplasm. It is composed of a mixture of structural proteins and glycoproteins. In bacteria, S layers are found outside of the cell wall, but in some archaea, the S layer serves as a cell wall. So remember, we talked about how some of the archaea have the pseudopeptidoglycan on the outside, creating a cell wall. Others do not have that cell wall. Some of those that we would say don't have a cell wall have this S layer, and then that serves as the cell wall. The exact function of the S layer is not entirely understood, and they're difficult to study, but they're suggesting that they may play a variety of functions in different prokaryotic cells, such as helping the cell withstand osmotic pressure, and for certain pathogens interacting with the host's immune system. So they're not entirely sure what the S layer, how it functions, and what it is specifically designed for, but they think it's to help with that osmotic pressure, again, because some of the organisms don't have a cell wall. It works as the cell wall, but then also interacting with the host's immune system. All right. So let's move on to our filamentous appendages. So many bacterial cells have protein appendages that are embedded within their cell envelopes that extend outward, allowing interactions with the environment. So these appendages can attach to other surfaces, They can transfer DNA, or they can provide movement, and we're going to talk about these different types. They include fimbriae, pili, and flagella. So first we'll look at fimbriae and pili. So fimbriae and pili are structurally similar, and because differentiation between the two is problematic, they're kind of used interchangeably. So, all right, so fimbriae commonly refers to short bristle-like proteins that are projecting from the cell surface by the hundreds. So when we see them, there are lots of them. Fimbriae enable the cell to attach to surfaces and to other cells, so they're little hair-like or bristle-like proteins that are sticking off of them, sticking off of the cell. So for pathogenic bacteria, adherence to host cells is important for colonization, infectivity, and virulence. So when we have a pathogenic bacteria, they're going to use these little bristle-like proteins to attach to a cell surface so that it can then go through the process of making copies of itself. So important for colonization, so it can hold on to something and make more copies of itself. Infectivity, so if it can hold on to the cell, then it can get into it and infect it. And then virulence, so that it can be protected and make more copies of itself to be more virile. Adherence to surfaces is also important in biofilm formation. So when we're talking about biofilms and creating that kind of community of bacteria that can't be easily washed away, these fimbria come in handy for that as well. The term pili commonly refers to longer, less numerous protein appendages that aid in attachment to surfaces. So again, they're longer, they can be attached to surfaces. And a specific type of pilus called the F pilus or the sex pilus is important in the transfer of DNA between bacterial cells. So there will be this, actually it says here, so which occurs between members of the same generation when two cells physically transfer or exchange parts of their respective genomes. So what can happen is two bacteria, and I think they're showing it down here, two bacteria will form a pilus here, an F pilus or a sex pilus, and then what can happen is the DNA from one will be moved along this little tube and move into the other one. And that transfer of DNA is important because then it can give genes to another bacteria, and then they can have the same bacteria in their genome. So sometimes you can see this with transferring important genetic information to help continue living. They can live longer if they can transfer some of their genetic information. All right, so then we have flagella. So the fimbria, the pili, and now we have flagella. So flagella are structures used by cells to move in aqueous environments. Bacterial flagella act like propellers. So they are spiral filaments composed of flagellin protein subunits, and they extend outward from the cell, and then they spin in a solution. Propellers are a pretty good analogy. It kind of spins around in a circle, and that allows the bacteria to move forward. The basal body is the motor for the flagellum and is embedded in the plasma membrane. So let me see if I can scroll down here a bit. Yeah, we'll get there in just a moment. So a hook region connects the basal body to the filament. So we have our basal body, which is the motor that's embedded in its plasma membrane, and then we have a hook region that connects the basal body to the filament. Gram-positive and gram-negative bacteria have different basal body configurations due to the differences in cell wall. So if it's embedded in the plasma membrane, you can imagine we have a difference between gram-positive and gram-negative since they have different plasma membrane compositions. Not necessarily plasma membrane compositions, but the peptidoglycan layers. So different types of motile bacteria exhibit different arrangements of flagella. So a bacterium with a singular flagellum, typically located at one end of the cell, which is polar, is said to have monotrichous flagellum. An example of monotrichously flagellated bacteria pathogen is Vibrio cholera, the gram-negative bacterium that causes cholera. Cells with amphitrichous flagella have a flagellum or tufts of flagella at each end. So spirillum minor, which causes spirillary, which is a rat bite fever. So cells with Lophotrichus flagella have a tuft at one end of the cell. So the gram-negative Bacillus Pseudomonas aeruginosa is an opportunistic pathogen known for many infections. So swimmer's ear, it's in burn wound infections as well. So they have Lophotrichus flagella. So flagella that cover the entire surface of a bacteria cell then are Peritrichus flagella. So let's scroll down here and take a look at this. All right, so we have our gram-positive bacterium and gram-negative bacterium. And you can see here's the basal body, okay, of the gram-positive, and you can see the different parts of that. You're not responsible for knowing those different parts, but you can see that basal body compared to over here. It's a little bit different. You can see it's different up in this area. So here's our cell wall. It's different here compared to here. So this is the cell wall here, and it's different, again, because remember we even have this outer membrane, the periplasmic space, the inner membrane of our gram-negative bacteria versus our gram-positive bacteria that has the inner membrane and then the cell wall. So we have our basal body here, and then we have our hook, this part right here, that's going to connect it to our filament for the flagellum. And then here we have our basal body here, and then the hook, and then our filament for the flagellum. All right. So here we can see the differences. So our monotrichus, that has the one here, one, mono meaning one. Amphitrichus, many here. We've got one here and then one on the other side. Lophotrichus, which is the tuft that they were talking about, so a tuft on the side there, and Peritrichus, which is often where we see many of them all around. So directional movement depends on the configuration of the flagella. So you can imagine this would be going this way. This can be going different ways. Bacteria can move in response to a variety of environmental signals, including light. So phototaxis is movement based on light. Photo meaning light, taxis, movement. Magnetic fields, magnetotaxis, magnet, and then movement. Using magnetosomes, remember we talked about those. And most commonly, chemical gradients, which is chemotaxis. So chemo for chemical, and then movement based on chemicals. Purposeful movement toward a chemical attractant, like a food source, or away from a repellent, like a poisonous chemical, is achieved by increasing the length of runs and decreasing the length of tumbles. So when running, flagella rotate in a counterclockwise direction, allowing the bacterial cell to move forward. In Peritrichus bacterium, so Peritrichus meaning the one that has the bacteria all around it, the flagella are all bundled together in a very streamlined way, which allows for efficient movement. So they'll kind of move all their all of their flagella and bundle them together so that they can move very fast. When tumbling, flagella are splayed out while rotating in a clockwise direction, creating a looping motion and preventing meaningful forward movement, but reorienting the cell toward the direction of the attractant. So rather than just kind of streamlining forward to try to move itself in a very specific direction, instead it can kind of tumble, reorient the cell to move in a different direction, so kind of turning in a way. So when an attractant exists, runs and tumbles still occur. However, the length of runs is longer, so runs moving forward there, while the length of tumbles is reduced, so turning, allowing overall movement toward the higher concentration of the attractant. So we'll be moving toward the attractant rather than tumbling to move directions. When no chemical gradient exists, the length of runs and tumbles are more equal, and overall movement is more random. So you'll see some forward movement, you'll see some tumbling to change directions, some more forward movement, and on. So here shows tumbling, so a clockwise rotation of flagella here, tumbling around, and then running or runs is all very specific. And we've got the clockwise rotation, or counterclockwise rotation of the flagella. So here you can see without the chemical gradient, here we see a run. The flagella is bundled, moving in a counterclockwise direction. It's going this way toward the attractant. The chemical gradient extends the length of the run, and then the run are bundled going counterclockwise, and is going to move this way and then tumble. So I kind of started down here, but this is probably where it's supposed to start here. So we're doing a run, and then clockwise rotation, tumbling, and then running again. So it's the way for the flagella to turn and change direction is this tumble. So we see runs and tumbles and runs and tumbles. And that's a little bit more random than the very specific runs that are going toward the attractant. All right, so that's the end of the 3.3 section. The 3.4 section you should be familiar with. This is something that's gone over in most intro biology classes and other specific classes. So make sure that you understand the rest of this chapter. Make sure you know that information. Again, I have posted other lectures that go over this in much more detail, and then I can also answer any questions in class or any questions via email based on any of these things.